What is up, my people? Well, I don't know if you're my people or not, but you're definitely people. And so, what is up, people? It is Wednesday, January 26th, 2022. We are reaching the tail end of January. This January went by pretty fast, but I'm glad it went by in a way that allowed me to start cutting my teeth and recording these videos for eternal life extension, which is really important. And I'm starting at the very beginning. So I did take a detour for this last week into hemoglobin starting on Saturday because I don't know, I'm a big exercise proponent, and hemoglobin, oxygen capture, carbon dioxide release, they're very important parts of human existence and animal existence and oxygen breathing species existence. So the cycle that exists in people is important to understand, and at some point might be important to compare to other animals that don't have the same situation going on. Amphibians, up to amphibians, I think most of them have some sort of hemoglobin, have the same cellular respiration and cardiovascular, have similar cardiovascular and cellular respiration mechanisms as people, or homologs, but at the same time some species do not. Anyway, going down a tangent there. So yes, let's get started. Yesterday I talked about the Bohr effect to an extent. So basically the Bohr effect is this here. I have this diagram here, and that's basically an increase in the affinity, a decrease in the affinity for oxygen as carbon dioxide binds to hemoglobin, and also as H plus ions bind to hemoglobin at specific amino acid sites along the chain, the hemoglobin, the beta side chains of hemoglobin, so at the beta subunits. So this is important because we want the hemoglobin to be able to hold on to this oxygen less tightly when we're exercising. So the Bohr effect is important while we exercise, right? Like, just think about Bohr effect, exercise, right? So more CO2 is held on to, more oxygen is released to tissues that need that oxygen, and then your ability to survive while exercising increases to an extent until, for example, there's too much lactic acid buildup, things like that. So that's the Bohr effect. And again, the details of how that works, it's very much on the amino acid protein interaction kind of discussion that we talked about yesterday, with how salt bridges form and kind of stabilize a molecule, so oxygen is not, the hemoglobin molecule, so oxygen is not needed as much. So it's really like a R state, forced R state conformation change that occurs because of this increase in CO2 binding and hydrogen plus ion binding. So it's important that both of those bind, and we'll see why. So one thing to note is that we have to talk more about respiration itself. So what happens when, jump in a little back, what happens when CO2 enters your red blood cells? So we have to know that because CO2 enters your blood, right? Because it's within your blood space already. It goes to your blood plasma. It doesn't really dissolve very much in your blood plasma because it's a nonpolar molecule. It's not charged, and about 10% stays in your blood plasma as CO2 gas, right? Most of that CO2 becomes bicarbonate, and that's very important. Bicarbonate is HCO3 right here, right? So this is important because this is the dissolved form of CO2. Again, we'll talk about how that occurs, but what happens is CO2, even though some of it dissolves in the blood as gas, most of it actually travels through your cell membranes into your red blood cells. And CO2 is a small molecule, nonpolar molecule, so it has no problem traveling across your cell membrane, your red blood cell membrane specifically in this case. And as it's as it enters, there's an enzyme called carbonic anhydrase, right, which has this structure right here. The equation right now is more important than the structure. Carbonic anhydrase converts CO2 and water to carbonic acid. And carbonic acid then disassociates to bicarbonate, which is this, right? And I always thought bicarbonate is weird because, bicarbonate, there's no two in here, so it only looks like one carbon dioxide, or one carbon and three oxygens with a negative charge. So, but anyway, whatever. This bicarbonate ion is the dissociated version of the carbonic acid. So once that occurs, you have H plus ions and bicarbonate floating throughout your system and throughout your red blood cell. And what happens is, eventually, most of the bicarbonate is expelled from your red blood cell through a channel, an ion channel on your red blood cells membrane, right? And so that's basically, you have it through an interaction through these channels here. Now, this right here is important because your red blood cells need to get rid of this bicarbonate waste product, which is essentially carbon dioxide. How does it do it? Well, it does it through electrochemical means, and that bicarbonate ion is negatively charged, right? So what has to happen? So there has to be a balance. As you're losing more negative charge, more positive charge builds up in the cell. The cell must maintain charge neutrality for several reasons, right? So in order to maintain that charge neutrality, this happens quite often through the rest of your body. This red blood cell has a ion channel which takes in chloride. So the chloride increases within your red blood cell, and the bicarbonate escapes from your red blood cell. And essentially, there are reverse channels to remove chlorine ions and whatnot. But this is called the chloride shift because you're exchanging your bicarbonate and expelling it into wherever, your alveolar space, or wherever the bicarbonate goes after it's expelled from your blood cells. So that was a quick detour. So now, why is the reverse effect of the Bohr effect, which is what I'm about to get to, I touched on a little yesterday, called the Haldane effect, important? So the Haldane effect is really considered the more important of the effects, because while the Bohr effect occurs and facilitates more efficient exercise, the Haldane effect is absolutely important for getting rid of waste product or carbon dioxide from your system. Okay, so this is kind of like the diagram showing how it's essentially the reverse reaction, right? So you have deoxygenated hemoglobin and oxygenated hemoglobin, right? And that's the interaction between both. Interaction between hemoglobin and oxygen is really what facilitates both effects, right? So remember, the Bohr effect is the absorption of H plus ions and carbon dioxide, the connection of that onto the hemoglobin molecule, which allows for decreased affinity for the hemoglobin for O2, which allows more efficient release into the tissues. But in the lungs, right, we have, by the way, these are the respiratory epithelium, as you can read, and this is the capillary epithelium, right? The respiratory epithelium really is what connects to the alveoli, right? And just to recall what the alveoli are, they're the air sacs that you learn about from when you're younger, that have capillaries surrounding them, and where the blood exchanges with carbon dioxide, and where all this occurs, where the Haldane effect really shines, essentially. So the blood is oxygenated, right? So we need to essentially be able to get rid of the carbon dioxide. So what happens is the H plus, as oxygen is taken up and oxyhemoglobin is created or conformed to, what happens is the decrease of H plus ions, affinity for H plus ions and carbonic acid occurs. So basically, we need to be able to absorb the H plus ions, and we need to be able to increase the amount of carbonic acid in the blood. So basically this reverse equilibrium equation occurs, right, and allows the increase of CO2 or bicarbonate, right, because we want the bicarbonate to be able to be expelled more effectively, right, through this chloride shift. So that's essentially what's happening here. The O2 is absorbing, um, is being taken up by the hemoglobin, right, and CO2 concentrations are increasing because bicarbonate concentrations increase, right, and thus in the blood plasma and in the blood cells, cytoplasm, and then that CO2 is then expelled from the cytoplasm into the bloodstream, into the blood plasma, and finally into the alveolar space, which is then expelled from your body. And this is happening normally, but the Haldane effect allows up to 50% more CO2 than would normally happen through your standard respiration process to be actively, via hemoglobin acting as a catalyst, to be expelled from your system. So that's very important. And just a quick review. These mnemonics here help to remember. Haldane, hemoglobin's affinity, Dane, carbon dioxide, right? I like that one. The Bohr effect, on the other hand, describes how carbon dioxide and H plus affects hemoglobin for oxygen. So oxygen concentration determine hemoglobin's affinity for carbon dioxide. So higher oxygen concentrations will decrease the affinity for carbon dioxide, thus allowing carbon dioxide to be expelled more effectively into your respiratory, across your respiratory epithelium as oxygen is taken in. So that's really important, right? This exchange that's going on here. So that's basically the Haldane effect in a nutshell. And next time I'll be getting over again to more molecular considerations in hemoglobin, but yeah, we should definitely take a tour into that. And there's so many good graphics to kind of describe how this works. The exchange is very important, because essentially we need the lungs to be able to expel CO2 effectively. Okay, and don't get confused between Bohr and Haldane effects. All right, see you around.